Hello, class. I'd like to first welcome you to Course 558, Neuroanatomy and Neurophysiology of Speech, Language, and Hearing for the Spring 2018 semester. It's a pleasure to be here with you, and I'm really looking forward to what we're going to be learning over the next few weeks. I'd like to first give a little bit of bio about myself and share with you where I come from. And then we'll go ahead and discuss the organization of the first half of the semester. And then we'll go ahead and dive in to today's lecture, Lecture 1. My name is Dorsa Beruchim K, and my background is in neuroscience. I studied neuroscience as an undergrad at UCLA and completed my PhD in neuroscience at USC, specializing in brain plasticity, learning, and stroke recovery. And I am passionate about communicating neuroscience with just about everybody. So I'm definitely excited to be here and excited to be discussing this material with you. I've outlined here the course of the next 8 lectures. Sorry, I jumped a little quickly. The next 8 lectures. And I want to share that a little bit with you so you know where we're starting from and where we're going to be going to step by step. I will first give you a general introduction of the nervous system today. And for Lecture 2, we'll go ahead and follow that up with the organization of the central nervous system and the peripheral nervous system. So we'll give you a little more detail there. We will follow up with the supportive structures of the nervous system. And by that, I'm including the meninges, the ventricular system, and the blood supply to the brain. We'll then tune in and give a general introduction of the motor system and sensory pathways. As you can see, we're starting out pretty broad and then tuning in to areas that are more relevant to motor and speech. In Lecture 5, we'll go ahead and discuss the neuromotor control of speech, and we'll first discuss the pyramidal pathways and follow that up with the extrapyramidal pathways. And then we'll conclude with a discussion on the visual system. And so we'll end with Lecture 7, which is vision, and then we'll have a review session before this section comes to a close. Before we get started with today's lecture, I want to first begin by discussing for a few minutes why learning this information on neuroanatomy is relevant to you as a speech therapist. So I know that from your perspective, gaining the knowledge and obtaining the information that you need to become excellent speech therapists is on the top of your priority list. And I'd like to share that coming from a neuroscience background, I think something that adds to you becoming an excellent speech therapist is the 3 elements that I've outlined here. And one that I hope over the next couple of months you feel more confident in. So the first one I've identified here is understanding current research. And as a speech pathologist, having the ability to be up to date with research that is being done in your field directly, and also in fields such as neuroscience and neurology that are relating and affecting and impacting how you practice is critical. And so as we discuss different fundamentals of neuroscience, I hope that you gain the skill to be able to also read literature and read research papers that are relevant to you and relevant to topics that you'll be specializing in. And I hope that that enables you to be a more competent speech therapist as well. The second point I've outlined here is improving your clinical practice. So hand in hand, having the ability to be able to understand research and the literature that's pertinent to your work, being able to work with your patients with an understanding of their underlying neurological situation. And so we'll discuss several examples over our lectures of potential situations and clinical cases that you might come across. And having a good basic understanding of neuroscience will enable you to have better comprehension over what your different patients are experiencing, what they may be having as other symptoms that are not as obvious or clear at first, and hopefully allow you to be able to treat them better. Thirdly, I believe that this could be one of the most meaningful aspect of gaining this neuroanatomy knowledge, which is you'll have the opportunity to educate your clients. And when they come in, you not only are able to give them help with gaining or improving their speech, but also be able to educate them about what they're experiencing, how they may have come to have the symptoms that they have and the types of impairments that they're experiencing, and also provide that knowledge to their family members and other professionals that you come across that you're trying to learn from or trying to give information over to be able to help them treat their patients better. And so I am glad to be here, and I'm looking forward to helping you through that journey. And I'm always available for any questions that you might have with regards to the material and how it might relate to helping you become a better speech therapist as well, and how it's relevant to you. So with that, we'll go ahead and dive into today's lecture. And today's lecture, as I mentioned before, will be a general introduction to the nervous system. And we'll go ahead and discuss the parts of the nervous system first today. And when I say parts of the nervous system, I really mean that very broadly, because the parts of the nervous system could be divided up in all sorts of ways. And one of the ways, or some of the ways, that it's mainly divided up, we'll discuss together. And I hope that that gives you a nice starting point into understanding how the nervous system is organized. Secondly, we'll go ahead and discuss some terminology that's used in understanding and learning and looking at the nervous system. And so we'll talk about neuroanatomical directions, planes, and general terminology there. And what I'd like to do is take that information to then be able to relate to the brain as a machine. And I want to just introduce to you an analogy that helps me appreciate the nervous system much more. And that is thinking about the nervous system as a car with parts and units and different building blocks. And recognizing that each of the units that make up a car are essential to allow for the car to function properly. And so what we will be doing is breaking up that car, or the nervous system, into its main components and also into the very units that create the building blocks that make up the systems that allow us to be able to see, to be able to smell, to be able to hear, to be able to walk, talk, and all the other remarkable abilities that we have that the nervous system allows for and provides that capability for. And so with that introduction, we'll then use the terminology that we learn to be able to discuss the different systems and also be able to utilize illustrations and images that bring to life the building blocks of the nervous system. So one of the main ways that the nervous system is broken up is taking the nervous system into central nervous system and the peripheral nervous system. And when we talk about the central nervous system, we're talking about the nervous system that integrates information to and from the periphery. And so this is from the outside world as well as the inside world, from our inside of our body. And that includes the brain and the spinal cord. I've depicted that here. The peripheral nervous system innervates our organs, our muscles, our joints, and our skin. And so, as I mentioned earlier, the peripheral nervous system then is what comes in contact with the outside world and the inside of our body, which would be the different organs and different muscles. And that includes the cranial nerves and the spinal nerves. And here I've depicted that here. So this illustration is demonstrating the location of the central nervous system and the peripheral nervous system. Taking a microscope, what I want to introduce to you now are the units that make up both the central nervous system and the peripheral nervous system. And that is the glorious neuron, which is here depicted as a drawing. So the neuron is the basic unit of the nervous system. And it could be divided up into 4 main components. Here we have the cell body. The cell body is the core of the neuron, which is similar to the cell body of other types of cells in our body. For example, cells that make up our skin and our other body parts. And the part of the cell body is where all the processing occurs, and where the nucleus is that houses the genome and the genetic material that makes up the role of that neuron and what it does and how it functions, and the specific thing that it specializes. The second is the dendrites, and the dendrites are these projections here. So if the cell body is this entire core in the middle, the dendrites are the projections that come off of the cell body from this end. And these dendrites' function is to take input messages from other neurons and send it down what we call the axon. And the axon is this projection here, this process coming out of the cell body. The axon is the main output projection, which then connects to other neurons via the synapse. And the synapse has specific terminals, which are basically ends of the synapse that connect to other dendrites or other cell bodies of other neurons. Looking at the nervous system, there is a distinction between recognizing the cell body as what we described and the axons that are the output projections that connect to other neurons. And what I'm showing you here is gray matter and white matter. And when we talk about gray matter, before I show you the difference in terms of the image that I've put up here, I want to first describe to you the components that make up the gray matter, and then we'll go ahead and talk about the image. The gray matter here is made up of mainly neuron cell bodies. They're also made up of other types of cells that are involved in supporting neurons, in specific glia and Schwann cells. Dendrites are also part of what make up gray matter, as well as terminals of axons and other types of fibers that are related to the nervous system transmission. I'm noticing here that I wrote unmyelinated fibers, and I'd like to just mention really briefly before we continue. I want to take a step back to share something with you. Here, the axon that is projecting out of the cell body has a protection layer called myelin, and that here is in yellow. And this myelination, this myelin, allows for faster conduction of signals that are being sent into the dendrites, to the cell body, and out through the axon onto the next neuron. And so this myelin here that is covering the axon allows for a faster transmission of signaling from neuron to neuron. And one analogy that that might help is thinking of a wire. A wire has coating around it to allow for the signal that's being passed through to be conducted through the area that it's being sent to and not spread out and leaked out of the wire that's carrying the information. Similarly, you could think of it as water that's going through a hose. So the actual hose that's covering the pathway that the water takes allows for that water to be sent through the hose versus leaking out if there was many holes, or if the hose wasn't a thick layer of plastic around it. And so in a similar fashion, you could think of the myelination of this axon as a way to protect and to help conduct the signaling that's coming down the axon in a efficient and high-speed form. So coming back here, the myelin that we were just discussing is fat. That's essentially fat that's surrounding the axon is white. And so here, white matter is really what's representing these myelinated axons. And so looking at this image here, I'm showing you a picture of a brain. And here, I'll first show you this one. Here it's let me orient you to the image first. Here it's an individual that's looking this way to the left. From my angle of looking at the slide, it's to the left, towards the text, let's say. And this is a sagittal view of the brain. And here we're looking at areas that are more gray and areas that are more white. And as you can identify, the gray area is what is making up the cell bodies mainly and the other elements that we discussed. The white area, also here is white, and here is white. It's just a lighter gray actually. That's the white matter, and that is identifying these myelinated axons, essentially the projections of the neuron that send information on to the next neuron. This image is of an individual looking from a coronal slice, and so imagine if this individual was looking straight at the computer. This is looking from behind him, and similar to what we looked at in this image, the lighter area is white matter, and so those are the axons, and the gray matter is essentially the neuron cell bodies as well as those other components that we mentioned. Here is an image of another form of imaging called DTI that allows us to look at axons and specifically to look at the direction that those axons are taking. And so here, color-coded is identifying the direction that those axons are taking within the brain. And what we're seeing are all those connections from different regions within the brain. And these are the white matter tracts. We'll talk a little bit more about what tract means. We'll discuss a little bit more about sagittal, coronal, as I discussed before. But that's essentially the division of gray matter and white matter. Again, just summarize, gray matter being mainly cell bodies as well as some of the other elements we mentioned, and white matter essentially being those myelinated axons that connect neurons to each other. When we're talking about groups of neuron cell bodies, one way that is generally used to name these cell bodies is dividing it up in central nervous system and peripheral nervous system neurons. So when we're talking about a group of neurons of the central nervous system, that's called a nucleus. And when we're talking about a group of neurons in the peripheral nervous system, that's called the ganglion. And here I'm showing you this image that contains real neurons from the thalamus, which is an area of the brain we'll talk a little bit more about later, that have been filled with a fluorescent dye and viewed through a microscope. And what we're seeing here are a cell body, also called a soma, dendrites, which are these long multiple projections that are around both this cell body and this cell body of both neurons, and the long axon that's connecting this neuron cell body to the dendrite of the next neuron. And so here I've also provided another image to make sense of this a little bit more. And I'll go ahead and point out the dendrites of this neuron are right here. The cell body of the neuron and the nucleus, which is where the nucleus is in the cell body, is right over here. And the long axon that then projects onto another neuron and another neuron here as well. And the ends of the axon, which we're calling the synaptic terminals, are also specifically look like buttons and are then called terminal boutons. I think it's French, if I remember correctly. So excuse me if any of you are French and can say that much better than I just did. But this is the specific area that connects and contacts the next neuron. And so this entire region that includes the terminal of the axon from this original neuron, as well as the area that receives the connection from this original neuron, which is here the postsynaptic receptor area. This entire region is called the synapse. And just to be comprehensive in this, as what we're talking about here, the synaptic cleft is the region in between the first neuron and the second neuron. I've highlighted in blue the ones that are essential for our discussion, but I did want to provide you a little bit more information for those that might be interested. The terminology for bundles of neuron axons here, I've included, again, similar as before, a division for central nervous system axons and peripheral nervous system axons. So when we're talking about central nervous system axons, we call that a tract. And so here I've provided an example called a representative of the cortical spinal tract. And so again, here's an image of a DTI image, diffusion tensor imaging, which allows us to look at axons in the brain and specifically look at directions of axons in the brain. And I'm looking for my mouse here. And this is allowing us to look at axons that originate in the motor cortex and are sent down to the spinal cord. And this is called the cortical spinal tract. We'll later discuss this in depth, but I want to give you an example of a set of axons that are in the central nervous system and what they may look like. In the peripheral nervous system, as we discussed, a group of axons are called nerves, And so here I have identified the hypoglossal nerve, also known as the cranial nerve 12, which controls the tongue muscles. So here in yellow is that hypoglossal nerve, and it's innervating the muscles of the tongue. Before we move on, I just want to take a step back and also give you a couple of examples for when the central nervous system nucleus and also peripheral nervous system ganglion. And so, for example, here when we were discussing a group of neurons in the central nervous system, we could take the example that's here, which is a couple of neurons from the nucleus of the thalamus, which is part of the central nervous system. And an example of ganglia would be the spinal ganglia or the dorsal root ganglia, which contain the cell bodies of sensory neurons entering the spinal cord at that region. Another brain division that I wanted to introduce you to is divided up into telencephalon, diencephalon, the brainstem, and cerebellum. And so this image here is one that I want to get you acquainted to. And the diencephalon, actually, let's start here. Yes, the diencephalon here is what includes the thalamus and the hypothalamus. Okay. The telencephalon is mainly the two cerebral hemispheres, as well as the basal ganglia and the limbic lobe, which is a combination of a few different regions of the brain that we'll discuss a little bit more later on. The brainstem is made up of the midbrain, the pons, and the medulla. So it's this section here. And the cerebellum is this beautiful piece of the brain, and I'll tell you why it's beautiful when I show you some of the histological images of the cerebellum, which are made up of very elaborate branches of dendrites. The neurons in the cerebellum are like arbors. And those are the main divisions of the brain. And I've also depicted here the spinal cord, which is, of course, part of what connects to the periphery. What I'd like to do next is discuss with you how the brain looks, which is a lot of wrinkles. And these wrinkles have very sophisticated names. These wrinkles have grooves, which are right over here. And as you can see, they're right over here as well. And they also have folds. And these folds are called gyri, and the grooves are called sulcus or sulci. When we have large grooves, we call that a fissure. And so we'll talk about how these come in to allow us to be able to divide up the brain into more functionally relevant regions. But I'll just go ahead and, as we're looking at this brain, identify one major sulcus called the precentral sulcus, which divides up the precentral gyrus and the postcentral gyrus. And here, the central sulcus. Sorry, let me take a step back. This is what I wanted to show you. The central sulcus, which divides up the precentral gyrus and the postcentral gyrus, which will be important when we discuss motor and sensory systems. This is definitely my favorite division of the brain, and it's because it's the functional division of the brain mainly. And that is by looking at the four lobes of the brain. Some of you may have heard about these different lobes, maybe heard a little bit about what they do and how they're important. And so I want to take a moment and discuss that with you a little bit. Here in blue is the frontal lobe. And this frontal lobe is involved with processing, motor planning, execution, as well as a lot of different language abilities. And as I'll mention in just a second, it's not the only area that's involved in language abilities, but is critical for it, as well as executive functioning. And so a lot of planning, a lot of critical thinking, a lot of higher order functions and behaviors and skills involve the frontal lobe. The second, here in pink or salmon color, is the parietal lobe. Sorry for this typo, I just noticed it says parietal, it should say lobe, parietal lobe. And that area processes sensation, including pain, temperature, and touch, and plays a role in language abilities as well, and is also essential in processing spatial information. So as you can see, so far with these two regions that I've mentioned, there are several connected functions that those areas are involved with. And it's not a sole like individual function for one or the other, that there's a lot of connection, a lot of overlap, and a lot of collaboration between different areas of the brain. Another region, the third of the four that I'm going to be discussing, is the temporal lobe. And here we have that in green. And this region is highly involved with processing auditory information, as well as involved with semantic memory. And we'll talk about an example of a clinical case where an individual lost a portion of his temporal lobe and lost a whole lot of memory, but specifically memory that involved things like recalling the city that she was born, or the place that, it's a he actually, the place that he visited last year, or what the capital of America is, or the color of the shirt that he was wearing last week. And so these types of memories are called semantic memories. And that's in contrast to memories that are more about being able to recreate a behavior such as riding a bike, or picking up a skill that doesn't require you to declare that information. And so in other words, called non-declarative memory. And so specifically, we're talking about semantic memory here. And the fourth, that's depicted here in purple, is called the occipital lobe. And that lobe is the lobe that is very involved with visual information and processing of vision. And it goes from simple projection of the actual visual image, as well as second order and third order processing of that primary information. Another common division of the brain is Brodmann regions. And so the classification of areas of the cortex here are originally defined and numbered by the German anatomist Corbian Brodmann, and he based it on cytoarchitectural organization of the neurons. And so he looked at the cerebral cortex and used a method called the Nissl method to stain the cells that made up the cortex, and divided up the regions that had similar types of neurons. And so Brodmann published his map of the cortical areas in humans, monkeys, as well as other species in the early 1900s. And this classification has been sustained throughout the years, and other ways of adapting this type of classification has definitely been developed as well, but this is one of the main ways that we do look at the cytoarchitectural neuron types that make up the cortex. And so here, for example, I've identified a few that might be relevant to us. And so the primary motor cortex here is called area 4. The primary somatosensory cortex is areas 1, 2, and 3 that make up that region, as well as Broca's area and Wernicke's area, which are both critical for language, and specifically Broca's area for language production, and Wernicke's area for language comprehension. And the primary auditory cortex as well, which is area number 22. The cerebral connections that are being made within the brain can also be categorized into 3 main ways. And so the commissural fibers are one type, which connect a portion of one hemisphere with the other hemisphere. And so an example of that is the corpus callosum, which does just that, connects the left and the right hemisphere. And these are the axons that connect with each other. A second type is the association fibers, and these association fibers connect different portions of the same hemisphere. So for example, the arcuate fasciculus, and I'll break that up a little bit to make more sense. Arcuate fasciculus actually means curved bundle. And this is the fibers that connect the caudal temporal cortex and the inferior parietal cortex to locations in the frontal lobe. The third type are the projection fibers, and these are fibers that connect the cerebral cortex to lower levels of the nervous system. And so an example that I've mentioned here is the corticobulbar fibers. And as the name implies, cortico meaning it comes from the cortex, and bulbar meaning it goes to the bulbar. And that is a tract of white matter pathways that connect the motor cortex in specific to the medulla, which is part of the brainstem's, also known as the bulbar. Now that we've discussed a little bit about a general overview of the nervous system, what I want to share with you now are key terms that are used to provide direction and orient us to images of the nervous system. And so on the right-hand side, I've included 4 main images. And I'll go through them. But what I want to first start off with is taking the nervous system of a dog. And so looking at a dog, the rostral end of the dog is toward the snout. And so if you can take a look, this is the brain, okay? And the rostral region is the front towards the snout of that dog, which would be also in the front of that brain. This is the brain of a human, and so the rostral would be in the front. So you're looking at the brain as though this individual is looking towards the text, and the eyes would be here. And so rostral would be front toward our face. Caudal would be toward the tail. And so if you take a dog, this would be towards the tail. And since we do not have a tail, the caudal from our human brain would be towards the back of our head. Ventral would be towards the belly when you're looking at this image here. So ventral is down here. And again, if you orient the brain similar to how it would be placed in this image, ventral would be then towards the bottom of our brain, which is basically in other forms where our neck is. Dorsal would be towards the sky, and that is looking up here in the human brain and towards the sky also for the animal. And so similar in that way. And so a couple other terms that I wanted to share with you are medial, which is towards the midline. So this image here is from the very side of the brain, but if we took a slice down the middle, that would give us a medial towards the middle of the brain. And lateral would be the position that's farthest from the medial plane or farthest away from the midline of the body, so towards the sides. Just to also orient you here, looking at the brainstem and the spinal cord, rostral again would be towards the brain this way. Ventral, again, we're looking at the basic version or the basic form of the nervous system here, where the brain is located here and the face is on this end, and ventral then becomes the very bottom. And so ventral in this way for the brainstem would be, for us, it would be in the front. And dorsal would be towards the back. And rostral would be up, and caudal would be down. So here's an image of our body just to orient you to dorsal and ventral as well. I find that if you imagine the human body laying down with our face down, then it gives you a nice analogy to the basic form that makes up how these terms are applied. Another set of names are anatomical planes, and so this is how we section brains. And I remember when I first started off in a research lab, we were studying songbirds, and we were sectioning the brain in coronal and sagittal sections. And so that's how I learned about this, but I'll go ahead and describe you guys the main 3 types of sectioning. And so this will come in very handy when we're talking about different images and different brain slices, okay? And so the first I'm going to discuss is a sagittal plane. The sagittal plane is essentially if you take the section straight down the mid from the 2 hemispheres. And mid-sagittal would be smack in the middle of the 2 hemispheres, and sagittal would be at any end. And so this would be sagittal here, and mid-sagittal would be very towards the midline, towards the middle. Coronal would be if you take a slice dividing up the face or towards the face of the brain and the back of the brain. And so coronal here, again, that would be giving a slice down here or at any of the other parallel regions. And this is the kind of slice that would give you. A horizontal or transverse slice would be taking, as the name implies, a horizontal slice so that you're kind of separating the top of the head from the bottom where our chin is. And this is the type of slice that you would get here. And so just to give you a little more of an orientation, this is the cerebellum here, and this is the cerebellum here as well. So I want you to take a second and see if you can kind of envision this in your own brain. Not that I want you to section your brain, but just to kind of envision where your cerebellum is and how it would be divided if you did it from a sagittal section, a coronal section, as well as a horizontal section. What I've also included here is a transverse or cross, which are also another term for horizontal sectioning, the same type of thing. Here's an image of an anatomical brain as well as an MRI that shows us a clinical orientation of the brain. And so here, now I'm going to use some of the terms that we learned. So as we discussed here, we are looking from the top of the brain. Okay, so this is the dorsal side of the brain here. And sorry, I jumped a little bit. This is the left hemisphere and this is the right hemisphere. And this, take a second and try to guess what kind of a section that is. Give you a moment while you're thinking. So this is a horizontal section of the brain, and this is an MRI. So it's as if we took a slice down the middle from here and we sliced it down the brain here. These are another set of MRI images. I just wanted to help you get acquainted. This would be a coronal MRI. And here, again, similar to what we looked at before, this is also a horizontal section. Another set of words, set of terms that are used for orienting the brain. Anterior, posterior, superior, and inferior. They are terms that are used similar to rostral, caudal, dorsal, and ventral, but are different when we're discussing the brain. Essentially, we're talking about the nervous system above the brainstem and below the brainstem. And so here I've provided an image that helps me very much. And here's the brain. We got dorsal, which is superior, posterior, which is in the back of the brain, anterior in front of the brain, inferior, which is ventral, below the brain. We're talking about the brainstem here, dorsal's here, and ventral is here. And so this is a slice down across here, the brainstem, and also allowing us to look at dorsal and ventral sides of the brain, or of the spinal cord here, actually. And so I hope that that's also helpful. Last couple of things I want to just discuss is one of the things that's really critical about the brain is the fact that there is two hemispheres. And that division carries on throughout the rest of our body with our left side and our right side. And oftentimes we'll talk about unilateral divisions, which means that it's initiated from or affecting structures on one side of the body. And so if we're talking about a unilateral damage, it's only on one side of the body, for uni standing for single or individual, independent one. When we say bilateral, we're talking about relating to or having two sides. And so bilateral would be encompassing both the left and the right side. Ipsilateral identifies the same side. And so if, for example, there is damage to the brain on the right hemisphere, and there is also impairments on the right side, that would be ipsilateral, which is on the same side. Contralateral, in contrast, would be relating to the opposite side. And so if we're taking the same example, if there's damage on the right side of the brain and there's impairments on the left side of the brain, that's contralateral damage. And this will be essential in understanding a lot of the wiring of the nervous system and its connection with the limbs of the body. So we'll go ahead and end our first lecture here, and we'll continue on next week with the next part when we provide a little bit more information and go into a little bit more detail about the nervous system. And I hope that we get a chance to discuss any questions that you might have. I'm happy to answer any questions or any anything that you'd like more clarification on when we meet for the Zoom session. So have a great week. I'll talk to you soon. And it was a pleasure having a chance to discuss this material with you.